Warahmatullahi wabarakatuh. Good afternoon, everyone, and a warm welcome to the parallel session of the 4th AMBC in conjunction with the 3rd MCLS 2021 in room 1. Maybe we will wait for a moment to the audience to join this room. And please fill the attendance and evaluation from AMBC in this room. You can check to the chat box. Okay, once again, good afternoon, everyone, and a warm welcome to the parallel session of the 4th AMBC in conjunction with the 3rd MCLS 2021 in room 1. I'm Fatiha Khairunisa, a researcher at Research Center for Biomolecular Engineering, or BIOM, and I will be your moderator today. In this room, we will have some presenters in which divided into 2 sessions who will share their research work regarding medicinal microbiology topic. Before we begin the session, please allow me to remind you that each presenter must have 10 minutes to deliver their presentation. And we also suggest the presenter to use the virtual background and switch on the video while presenting. And then after 3 until 4 presenters, we will have Q&A session. So if the audiences want to ask, they also require to type the questions in the chat box. And then, okay, I will check the presenters for the first session. I will check. Mr. Udom Seuang, are you there?
Hi.
Yeah, okay. And then the second presenter is Ayu Eka Fatril.
Yeah, I'm here.
Yeah. Okay. And then the third presenter is Arif Nur Muhammad Ansari.
Yeah.
Yeah, okay. And then the fourth presenter is Sri Anggarini Rashid. Sri Anggarini Rashid, are you there? Okay, maybe we will switch the presenter, yeah. Okay, for make this session more efficient, I will let's begin with the first presenter. I will call the first presenter is Mr. Udom Seuang. He is from National Center for Genetic Engineering and Biotechnology, Thailand, with his presentation title C22 Podovirus infectivity is associated with intermediate stiffness. Are you ready to start your presentation, Mr. Udom?
Yes, let me share my and see if you can see it well. Okay, hold on, sorry. A bit, share screen, screen 1. All right, so do you see this well, my screen right now?
Yes, I can see.
Okay. All right, I think that's, hold on.
Yes, you can start now.
Okay.
10 minutes is yours, yeah.
Okay, I can start now, I think. Hi. So, yeah, my name is Udom, right? So I'm from National Center for Genetic Engineering, Biotechnology of Biotech, Thailand, which is part of NASA. And so I'd like to thank the organizing committee for this opportunity. As you will see shortly, that my work is not quite in the domain of medicine or microbiology, but I sincerely hope that you will enjoy my talk, and there may be something we can work, collaborate together in the future. This work was has been published in scientific reports last year, but due to the COVID situation, I never have a chance to present anywhere. So this is the first time I present in the conference. Okay. Hold on. All right. So you know phage very well. Bacterial virus or bacteriophage has become one of the most attractive choices for biocontrols, and especially for pathogenic bacteria in crops for agriculture. And because of three advantages, and I call it 3S. So the first S is specificity. So it's very specific to the host cell. And secondly, is the self-replication ability. It can replicate as long as the host cells are there. And thirdly, it is generally considered safe for agriculture application. So this we call 3S. And of course, there's a reason that why the phage is still not widely used in biocontrol when compared to other biocontrol agents. And one of the reasons is that the efficacy of phage biocontrol is variable, depends on many factors, and is poorly understood. So this study is one of the attempts trying to study another study more. And the one thing, sorry about that. One thing that I like to understand more is the stability. Okay, and when it talks about stability, let's consider it from structural point of view. Oh, by the way, I'm a physicist, so when I say many things, my biology background is very low, but I try my best. Okay, thank you for that. Sorry about that. So from the structural point of view, the outside of the phage is a protein. So that's capsid, right? And inside is the internal is the genomic material, DNA or RNA, or double-stranded, single-stranded. And both capsid and the genomes are very sensitive to the external factors like pH, salt, temperature, osmotic states, especially if this phage are in the environment, in the soil for biocontrol, they will experience all of this. Okay, so we ask a very simple question: what are the effects of these factors on the phage stability, on the structural components, right? And but this question, even though it's pretty simple, the scope is huge, right? What factor are you talking about? Blah blah blah. So we try to scope it down a little bit, try to address some of these aspects in this study. So, and the first thing I do is that the phage that we're gonna study is called C22 podovirus phage. Okay, so C22 is a phage that we isolate and discovered in Thailand, in the Chiang Mai, the northern part, and this phage is capable of lysing or killing Ralstonia solanacearum, which is the cause of bacterial wilt disease in chili, tomato, tobacco, potato, basically. But this one specifically for chili, I think. Yeah. So it's a soil-borne pathogen, right? So that's the phage that we can look at. And when we talk about factors, let's focus on two factors here: pH and ionic strength. But when we talk about stability, I am going to approach it from different angle because I'm a physicist, so I look at things physically. So we're going to talk about nanomechanical properties. So if you're not familiar with this term, just think when we talk about mechanics, we talk about things that are hard, are soft, right? Oh, this is hard, this is soft, that's mechanical. So nano, because the phage is very small, that's all. And basically, we will try to measure how tough, how hard, or how soft these phage particles are. And if you want to measure if something is hard or soft, it's pretty simple, right? You're basically gonna use your finger. And just like I have a soccer ball here, and I want to push, I want to see if it's hard or soft, I'm gonna push it, right? So that's exactly what we're gonna do here. We're gonna pretend that this phage particle is a soccer ball, right? And we immobilize it on some smooth surface, mica, glass, actually mica would be better. And then this cantilever behaves just like our finger that touch and push and indent these virus particles. So this machine is called atomic force microscope. Okay, and I'm going to use this machine to measure this property of C22 podovirus when I vary pH and ionic strength. Okay. All right. Wow, it's already 4 minutes left. Wow. Okay. That's so these some pictures. We can use this same technique to measure the to get some image as well. So A, B, and C are the AFM pictures. That this is a TEM. So nothing advanced there. And when we measure, and I try to push on this virus, we obtain something. We can measure the slope, and we obtain something called force distance. And by some calculation, if we take the force and divide by the distance, this distance is the distance that the cantilever push into the particle. Then we get something called stiffness, right? It's Newton per meter spring constant, blah blah blah. You've seen before. So by doing that, we can hold on. I think okay. We can measure the stiffness of virus using this machine, and once you start to measure this number, however, there is just one single value of stiffness. If I measure it 10, 20 times, you can imagine that there will be some variation of that number, right? You're not going to get like 1, 1, 1 all the time. That's not how it works. So, and you get distribution. We get distribution of these stiffness values of C22 protovirus, and we measure it in pH 6, 7, 8. We change and explain the salt component, right? And we do that, of course, and we see that there's some changing of this stuff, which is of this value, which is not surprise. I mean, it's supposed to change, right? We change pH, structure got to behave some way, but we also measure that. I don't have time to go into detail. The infectivity, right? Our assumption is that it got to do something with the virus. Otherwise, why would we measure it? So we look at the titer or the remaining phase that we store in the same pH and ionic strength as we did in the AFM measurement, and we try to compare if we can see, and with a lot of control experiments, of course, we try to, and we found some correlation. We found that we found that the phase is the high stability, the number of phase that is remain after 6 months is actually correlated to the stiffness when it's in the middle or intermediate stiffness. Or in other words, phase shouldn't be too hard or too soft, which is a bit surprised, right? Because, well, you may think that physically you want phase to be really, really hard, that way it can protect itself, but actually not. It cannot be too hard, too soft, kind of intermediate. So that's what we have. That's essentially the core of the result that I have here. So I have 1 minute left, so I don't want to go over time. I think I get to the conclusion. Hooray! So the conclusion is that we measure the phase. We can measure with phase the stiffness, this nanomechanical property of virus, and we have done it for cells, for virus, for fungi. Actually, that's what we do. And so that's my kind of what I like to do. And then we try to associate these properties to the function. I mean, we know there's something called structure-function relationship, but that structure got to change and express and accompany something, right? And we believe that nanomechanical property might be that thing that bridge between the structure and the function of biosample. And lastly, when we talk about something in the middle, actually virus have this behavior before. It's called viral metastability. So simply speaking, this means that virus should be strong enough to protect itself from the surrounding, but should be flexible enough to interact with the host cell. And we think that this is some kind of adoption of viral metastability. And with that, I think I've come close to at 10 minutes. So I'd like to thank you again for this funding and then blah, blah, blah. Well, they my institute. And oops. And if you have any question, you can contact me. Thank you very much.
Thank you very much, Mr. Udom, for the great presentation. I think it's very interesting because you have published your work in Nature. Okay, then we move to the second presenter. The second presenter is Ms. Ayu Eka Fatril with the work about molecular analysis of Aspergillus spp, isolates from probable chronic pulmonary aspergillosis patients using beta-tubulin gene. She is from Department of Parasitology, Faculty of Medicine, Universitas Indonesia. Okay, for Miss Ayu,
Yeah.
are you ready? Please share your presentation and start.
Is this clear?
Yes, it's clear.
Okay. Thank you for moderator.
Please start your presentation.
Oh, okay. Thank you for moderator to give me a time. So I will try to present our research about molecular analysis of aspergillosis species isolate from probable chronic pulmonary aspergillosis patient using beta-tubulin gene. So in introduction, first we know CPA or chronic pulmonary aspergillosis and pulmonary tuberculosis is infection in pulmonary. So what different from them? CPA or PTB is different. Called CPI because cause as aspergillus, usually Aspergillus fumigatus, and PTB or tuberculosis caused Microbacterium tuberculosis. That's bacteria. In mortality rate, CPI have a high, quality high, 20 until 50%, and some with treatment CPI using triazole, amphotericin B, and caspofungin. In the case, around 1 million people in the world have a CPA as sequel to TB, and Indonesians have a quality high CPI case. So that's a big problem. No, PTB and CPI had the same symptom. So many doctor on analyze confused because that. So many make misdiagnosis about CPI and they said CPI as PTB. And aspergillosis is infection because aspergillus and species infection like Aspergillus fumigatus, Aspergillus flavus, and Aspergillus niger. And that variant have so many different or variation in clinical manifestation, virulent factor, antifungal response. So in so many variants, we need to species identification. Usually in laboratorium, we just using morphology or phenotypic identification, just macroscopic or microscopic. So it's not enough to different that. So we want to using molecular like sequence analysis. Usually sequence analysis we just using ITS for identification, but in some papers so using in Aspergillus, ITS is not optimal again. So we chose other primer, we chose other gene to space to identify. So we're using beta-tubulin. Beta-tubulin gene is a gene called tubulin protein to cellular process. Tam et al. Said beta-tubulin can to identify Aspergillus flavus, Aspergillus nominus, and Aspergillus tamari. And Nasri et al. Said too, using beta-tubulin gene can identify unique pattern from 6 clinical important Aspergillus species. And beta-tubulin 2 as a DNA barcode. All about benefit in beta-tubulin, but we saw in Indonesian data, we still have information. We still didn't have information. Information is still available about identifying using beta-tubulin, especially for CPA patient. So we want to research about that in study aim. First, we want to evaluate beta-tubulin as DNA barcode, and we want to confirmation Aspergillus species molecular identification using beta-tubulin from probable CPA sputum samples. For material, we using 31 isolate from sputum sample of PTA patient. We using primer BT2I and BT2B, and method we are using a PCR single product. And result, first we want to compare phenotypic identification and molecular identification. In phenotypic identification, we just find 3 species. It is Aspergillus fumigatus, Aspergillus flavus, and Aspergillus niger. But so in molecular identification using beta tubulin gene, we find 7 variant as Aspergillus species. First, fumigatus, flavus, tamari, niger, tubingensis, welwitschii, and bruno facialus. So with this, we found so many variants in here, and many variants variant of Aspergillus 2 can to help we are to using about clinical or another antifungal response. And we try to analyze the sequence using multiple alignment in 29 sample. In here, we saw in the red, in the green line, and I'm so sorry, is clear the most or no? Moderator.
Not really clear.
Oh. In here, still not clear.
Yeah, it's okay. It's okay.
Okay. Yeah, this pointer in the green line is so many variant same variant base. So it meaning using beta tubulin we can to cite beta tubulin is conserved genes, and we try to analyze in a 7 species in Aspergillus Aspergillus species. In a red box, we see in red box is many variant based, variant based. So very variant based in we can to meaning that beta tubulin gene can to identify, can to discriminate Aspergillus species and in 7 species. It's a meaning that to using beta tubulin gene can to hide interspecific variation. So we try to analyze sequence of Aspergillus fumigatus for PTB samples. Only Aspergillus fumigatus. In here, we saw so many same base in here. So it's meaning using beta tubulin gene have a less intraspecific variation. And we try to one section is in Aspergillus species is a flavi section. In flavi section had a member like Aspergillus flavus and Aspergillus tamari. In here, we saw in the red box different base sequence. So it's meaning using beta tubulin gene can identify until species in same section. So in first in phenotypic identified, we just said is Aspergillus flavus, but in molecular identified and sequence identified, we can saw it not just Aspergillus flavus, but it Aspergillus tamari too. And we try to analyze in another section is nigeri section. In nigeri section have a member like Aspergillus niger, Aspergillus tubingensis, Aspergillus welwitschii, and Aspergillus bruneocoeleus. In here we saw another so many variant base in here. So Aspergillus beta tubulin gene and have interspecific variant of in the nigeri section partly high, but not for Aspergillus niger and tubingensis. Why we can to not enough to discriminate or identify about niger and tubingensis? In another table, we can saw in here what in similarity rate is so it's quite high.
Minutes remaining.
Okay.
Are you?
Okay, thank you. It's 82.5%. So sometimes it's be little trouble to identify molecular. And we try to gene mapping beta tubulin. In here, it's all Aspergillus flavus or Aspergillus species have 4 exon and 3 intron. the number of segment of axon is similar in all species with size do not differ too much, and conserved axon are good location for primer attachment. Meanwhile, the intron between axon sequence serve the source of variation. So beta tubulin is a conserved gene, less intraspecific variation, and strong intraspecific variant. It is quietly as a DNA barcode. So the conclusion we said the beta tubulin gene show low intraspecific and high intraspecific variation. They might be suitable for Aspergillus species identification of part of CPI diagnosis strategy. Thank you. I back to moderator.
Okay, thank you so much, Ms. Ayu Eka Fatril, for the nice presentation. And then we move to the third presenter. He is Mr. Arif Nur Muhammad Ansori. He will present research work about a therapeutic potential of Newcastle disease virus against cancer, a virotherapy approach. He is from Universitas Airlangga. For Mr. Arif, are you ready?
Yes, of course.
Yes, please share your presentation.
Okay. Hello, everybody. It's an honor to present here with the topic a therapeutic potential of Newcastle disease virus against cancer of virotherapy approach. Here's the paper with my colleague from University of Surabaya. Okay, to begin with, I will talk about the cancer. So the WHO stated that in 2015, there are 8.8 million deaths caused by cancers, and also in Indonesia, based on the data in 2014, there are two types in men and women based on the gender. So the top three are lung cancer, liver, and colorectal cancer in men, and breast cancer, cervical cancer, and lung cancer in women. And then we had WHO predicted in Indonesia in 2030 increase up to 7 times. Cancer is an important cause of mortality, and the lack of the efficient therapy, such as chemotherapy, radiotherapy, immunotherapy, and so on. And then new approach is oncolytic viruses or virotherapy. And the Newcastle disease virus is a virus infected avian and other species. So this is the genome of Lasota strain. Lasota strain is common for vaccine construction against the Newcastle disease virus. So this is the structure of the virus. There are various proteins such as matrix protein, nucleocapsid, phosphoprotein, and L protein, HN, and fusion. So the fusion and HN protein is important for induced immunity. So the size of the genome is 15 kilobase, and the virus diameter around 300 nanometer. So this is the mechanism of infection. The first binding to the cells, and he checked the mechanism, cellular mechanism, and until the development of new virions, and then they will infect other cells. So this is the epidemiology of NDV. NDV infected various species of avians, such as chicken, duck, and goose, and so on. There are so many disadvantages, such as economic loss and virus impacts. Uniquely, the virus first identified in Java in 1930, based on the history. So we already mapping the Newcastle disease virus from 1976 to 2019, based on NCBI database. There are so many type of the virus, such as lentogenic virus and phalogenic virus. Phalogenic virus is a virulent one with the cleavage motif sites like this. Okay, we go to the virotherapy approach. So a bibliometric study stated that always there are increasing number of papers about the oncolytic viruses based on Mozaffari Nejat et al. In Virology Journal. And here's the data. And then this is the institution and the journal based on their data. And also the authors who virotherapy approach or oncolytic viruses. Okay, so why NDV can be used as a cancer therapy? Based on Kalyana Sundram et al. They stated that because of non-pathogenic in human and rapid viral growth in cancer cells and oncolytic activity, and then increased performance of immune cells, so the ability of the virus to induce cytokine synthesis, and then virus increase expression of viral antigen on the surface of cancer cells, and then capability of the virus bind to the surface of the cancer cells. So here is the schematic diagram. We can see here the virus, oncolytic virus, Newcastle disease virus. Actually, oncolytic virus, not Newcastle disease virus. This is one of the example. And then the normal cell. In normal cell, they blocked and cannot replicate. But in cancer cells, they can replicate until 1,000 times more rapidly than in normal cells. And then the cancer cell disease and the new virion release and infect the other cells. Okay, Ginting et al. Studied about Lasota strain, the common strain for vaccine development, and they present that this is the the viability in normal cells, and the right one for the tumor cells, such as U87 and then MGF7 and so on. The viability of the cell is decreased. And then another scientist using AF2240 strain or phalogenic strain from Malaysia, originated from Malaysia. They unlocking the extrinsic pathway and intrinsic pathway until the apoptosis process in cancer cells. So it will be very great if we can explore more about the NDV in Indonesia to test against the cancer cells like that. But there are no researcher in Indonesia working in NDV as ferrotherapy. This is the data from Kayana Sundram and Yurchenko in PLOS One published the wild type strain from Russian Federation. We can see the tumor volume in mice decreased than mice treated with PPS. You can see the dark one, the blue one here after the ferrotherapy, start of ferrotherapy, and 15 days after NDV ferrotherapy. So we can see here in in vivo study and the histology. Actually, the Yurchenko et al. Not only isolate one virus, but there are many viruses isolated, but Yurchenko from Russia, and then they screened the isolate with high activity in oncolytic virus. Okay, so the conclusion in summary, we demonstrate the promising oncolytic virus against various human cancers, and therefore we suggest that NDV originated from Indonesia can be tested in vitro or in vivo in the near future, because it will be very great for scientific in Indonesia. So this is acknowledgement and reference based on our latest publications. And thank you. Steve Jobs said that I think the biggest innovation of the 21st century will be at the intersection between biology and technology. Okay, thank you.
Okay, thank you, Mr. Arif, for the great presentation. And then I will call the fourth presenter in this session, Ms. Sri Anggarini Rashid. Are you there? Ms. Sri Anggarini Rashid. Okay, if you don't attend to this session, maybe I can switch to the second session. And then let's move to the Q&A session. So we will discuss about the three presentation from our presenters. So if you have any question, you can type your question in the chat box, or you can open your mic to ask directly to the presenter. I will check to the chat box. Oh, okay, I think there is a question from Mr. Rizky Danang, but Mr. Udom have answered that question. But I think I can repeat the question so Mr. Udom can explain again to the audience so we can get more clear about your explanation. Okay, from Rizky Danang, nice presentation, Dr. Udom. Can you explain again how the relation from titer and days to flux-based infectivity?
So yeah, we found that the remaining higher, we found high remaining titers is correlated to the medium value or medium range of stiffness, as I showed at the end. But to get to that conclusion, actually there's a lot of control experiments, because you can imagine that the titer that we measure from the PAC assay, that is the result from the phase host interaction. And there's so many things that can happen. I mean, stiffness can be one of them. There can be many things. So that's what a lot of those experiments has been done to measure, and then some statistical analysis to show on that. So that's the work. Yeah. So of course, and then the next thing is, why is that? And we think that is come from the structural rearrangement, because the stiffness that they measure directly come from the structure of phase. But in what way? And there's several studies that have been started to show what exactly this mechanism of phage or even virus that correspond to these factors. Yeah, but we're still doing that. Thank you.
Yeah. Okay, Mr. Udom. Is there any comment from Rizky Danang?
It's enough. Thank you, Dr. Udom.
Yeah, okay. Are there any questions from other presenters or audience? Please don't hesitate to ask question to the presenter. Okay, maybe I would like to ask to Mr. Udom, with your research work, what kind of application in the therapeutical area?
Oh,
Maybe is there any application
that's a
about
good
your
question.
research work?
So I tell you what I've been done, because what we just published, like actually last month, is that we start to measure stiffness of cancer cell. So we found that, well, where the I don't remember exactly, but the cancer cell actually start to get stiffer. It's getting harder when compared to the normal cell. Of course, that number or that information probably cannot really directly apply for therapeutic utilization immediately, right? But I think we try to understand this behavior as a part of how the cell actually rearrange their cytoskeleton in correspond to even drug resistance, or what type of the different type of cell, how they different respond to different stimuli. So to answer your question exactly, in that we try to basically understand this biosystem more, because they are so complex, they are so amazing. At least from my perspective, I really love the biology, even though I don't understand much. But I think there's a lot more that we can learn, especially from different perspectives. In that way, we can combine the genomic information, cell-based technology, and even structure, and even this nanomechanical, and understand it more. Maybe they can bridge up to something. I would say more like indirect. That's not really the exact use, but when we try, I think it's fun.
Oh, okay. So your work will you can develop your work to apply it in therapeutical area, right?
Yes.
Okay, and then I will back to the chat box. This is a question. There is a question again from Rizky Danang to Miss Ayu. Nice presentation, Dr. Ayu. Where are you still researching phenotypic when molecular is more accurate to identify Aspergillus? Miss Ayu, you can answer that question.
Okay, thank you to your question, Mr. Rizky Danang. I will try to answer that. We want to compare the phenotypic result and molecular identification, because usually in laboratory normally only using phenotypic. So we want to prove, or say in a another method where the best method to identify sample like aspergillosis. And maybe why we still compare, because that not all laboratory can to using molecular study too. So with the research, we hope will help another laboratory to think another way to identify aspergillus. Thank you.
Okay, thank you, Ms. Ayu. Is there any comment from Rizky Danang?
Okay, it's enough. Thank you for your explanation.
Okay, the third question is also from Rizky Danang. This question is for Mr. Arif. Dr. Arif, what's function for mapping virus based on year?
Okay, thank you, Mr. Danang. The answer is the mapping of the virus is on Jur. We can see the changes of the virus, maybe the mutation or pathotypic changes in Newcastle disease virus. In to develop vaccine, for example. But in the cancer therapy, like Yurchenko et al. Stated that they mapping the virus and also all the genome and identified the pathotypic, and they test in vivo and in vitro against the cancer cells, and also using an animal model with the cancer, like that.
Are you satisfied with the answer, Mr. Rizky?
Okay, thank you, Dr. Arif.
Okay, is there any questions from one presenter to another presenter maybe? Or from the other audience? Okay, maybe I have one question from Miss Ayu. Actually, how do you use the beta tubulin gene in the experimental of your research? How can you analyze the Aspergillus patients with your invention in beta tubulin gene? Are you use a PCR method or anything else?
We are using single PCR method. So maybe it's easy to using that. We're using primer with BT2A and BT2B as a primer. And in here, we try to get ampicon. So after that, we are sequencing. So in a sequencing result, we blast and we compare with NCBI to get identified identified species.
Okay, I understand. And then for Mr. Arif, you said that about in vitro and in vivo testing. In your conclusion, can you explain a brief explanation about how to test in vitro with your research?
Okay. Actually, in Indonesia, there are no isolate used or test for against cancer cells in vitro or in vivo. One study from Malaysia, the isolates origin from Malaysia, tested by various researchers in Malaysia and or outside Malaysia, and they conclude that the isolate is potent to prevent or maybe against the cancer cells. But in Indonesia, there are no research about that. Then maybe this is the new topic, and maybe I hope maybe in the near future, various researchers we can together to join for the research in oncolytic virus, especially for NDV.
Yeah. Okay.